this is the plant infested plant and this is the pest resistance plant. तो जो निमेटोड है, क्या निमेटोड नाम लिया था मैंने, क्या नाम लिया था? Melidogyne incognita. So it infests the roots of tobacco plant and it is also seen infesting the tomato and brinjal plant. And a new technology is developed that is called RNAi. The full form is said to be RNA interference, and that will help in preventing the disease. Now, how it works? What is RNA interference and how it works? And what is the technology? Now, RNAi take place in all eukaryotic organisms. So this is a common method of protection of plant. So plant protect themselves with the help of this method, and that's why this method is called as what? The method of self defense. So it is common method of self defense, okay, in eukaryotes. Now, to develop the pest resistance, silencing translation of specific mRNA. Silencing, the word is silencing, the word translation of specific mRNA. Now, if you see this is a double-stranded DNA which has a template strand, which has a coding strand. It undergoes transcription and forms mRNA, and from mRNA protein is going to be formed. So we are involved in this process where mRNA is going to transfer into protein. So we have to check the process of translation, okay. Silencing of specific mRNA due to complementary double-stranded RNA, a type of RNA molecule to enhance or inhibit translation of mRNA, binds to mRNA and prevent translation. देखो, तो हमें एक स्पेसिफिक mRNA को क्या कर देना है? What we have to do? We have to silence it. Okay, silence means it is not going to express, no expression will be there, okay. So no expression will be there in this, and because no expression, the nematode will not be able to live. क्योंकि expression नहीं होगा तो क्या नहीं बनेगा? प्रोटीन नहीं बनेगा, और प्रोटीन नहीं बनेगा तो then it will not be able to survive in absence of the proteins. Now, how to make this? Due to complementary double-stranded RNA, a type of RNA molecule to enhance or inhibit translation of mRNA, binds to mRNA and prevent the translation. तो हम यहाँ पर एक और what we will do? We will use the double-stranded RNA, and that will silence this. Source of double-stranded RNA, from where this RNA will be taken from, it will be complementary RNA, and it is a viral RNA. तो इसी का जिस part को हमें silence करना है, we have to use a complementary RNA from viruses or from transposons. What is transposons? Genetic element which replicate via RNA intermediate, okay. Then how it is done? Now we have prepared double-stranded RNA. Now this RNA is introduced into a cell. जैसे ये है तो ये है double-stranded RNA. The enzyme dicer, this is a dicer enzyme, is cleaving the double-stranded RNA in the middle of polynucleotide. तो यहाँ वो उसको क्लीव कर रहा है. Small interfering RNA, siRNA are formed. क्लीव कर दिया, फिर दोनों अलग हो गए, सेपरेट हो गए। सेपरेट होने के बाद सेपरेट करने के लिए, बनाने के लिए क्या ज़रूरी है? RISC। RISC मीन्स इट इज़ अ small interfering या RNA induced silencing complex. Okay, small interference RNAs are produced by RISC complex, which is called RNA induced silencing complex. So now the RNAs are separated. Single stranded RNA coupled with RISC and then binds to complementary mRNA. Now this will also help to bind one of them with the similar code of the gene that we want to silence, and it will make a complementary structure and it will target mRNA cleaves, और उसको क्या करेगा? It will break down this mRNA. So because the mRNA is broken down, no further reaction will take place. There will be no protein formation. Okay, तो हमने क्या कर दिया? What we have done? We have stopped translation. We have stopped translation process. Okay, and hence the target mRNA cleaves and translation is prevented. So in this way, because of absence of protein, the organism will not be able to survive. Using agrobacterium. Now how to do this? Using agrobacterium vectors. You learnt about these vectors. Nematode specific genes were introduced into the host plant. So what is the host plant? So the host plant is a tobacco plant. So it is introduced into the host with the help of a gene. The gene is introduced with the help of a agrobacterium vector. The introduction of DNA will produce both sense and antisense RNA in host cell. So when it reaches into the cell, it will produce sense RNA and antisense RNA. Now, since sense and antisense RNA are complementary to each other, double-stranded RNA that initiate, so this will become a dsRNA, and then it will work as we have seen in this. Where double-stranded will attach with a dicer, then it will cleave this, then dicer will be cleaved, then RISC complex is attached, which differentiate both the double-stranded RNA. Now we have single-stranded RNA. Then single-stranded RNA will get complementary with the mRNA. Yeah, this is mRNA, and then it will, with the help of the RISC complex, it will cleave the mRNA, and hence no protein formation will take place. Okay, so genetically what we have done, we have introduced the DNA specific to the host plant, and this DNA then will produce two different RNA, that are sense RNA and anti-sense RNA. Clear? When nematode infests the plant, now it is happening in plant, introduction of nematode genome take place in presence of double-stranded RNA, which silence specific mRNA of nematode, and initiation of RNA will take place. Okay, when nematode infests the plant, introduction of genome, nematode specific genome will take place, and the presence of double-stranded silence specific mRNA of nematode will initiate the RNAi. The parasite will not able to survive in the host which express interfering RNA. यानी जैसे as and when he infests the host plant, the cells that will have silence specific mRNA will reach into his body, and there will be interfering RNA seen inside the nematode. Okay, transgenic plant therefore got resistant and protected from the parasite. So this is a new technology that is introduced for the pest resistance and especially the nematodes, and that is Meladogyn incognita. Clear? So this was the complete structure and process of RNA interference. So let us see some more summarize or recapitulation. Several nematodes parasites a wide variety of plants and animals including human beings. RNAi is a gene silencing, ये word तुम्हें याद रखना है, that is what it is also called as gene silencing mechanism triggered by providing double stranded RNA that when ingested into insect can lead to death or affect the viability of target pest. Okay, this will enter into the pest and double stranded then will get separated into sense and antisense. Both of these they are since sense and antisense are complementary to each other, double stranded DNA double stranded RNA is formed. Okay, and double stranded then will enter into the host plant and with the help of a bacterium, and then it will work according to the steps that we have discussed. This is the complete pathway by which the RNA interference works. So double stranded now entered, it will cleaved by dicer, separated by risk, and then complementary made by the risk complex, and then cut into short fragments. Now, which nematode infests the roots of tobacco plant and causes a great reduction in yield? तो क्या आंसर दिया जा सकता है? We can give what is the nematode? Meladogyn incognita. Clear? This is the one. RNA genes are silenced using double-stranded RNA. Silencing of gene could be achieved through the use of RNA only, antisense RNA only, by both, or none of the above. So what should be the correct answer? C. From my view, it is C is the correct answer. siRNA are produced by transcription, DNA damage, RNA damage, or enzymatic reaction. So RNA formation take place with the help of transcription. The RNA mediated gene silencing includes binding of siRNA to form a complex with the target gene called RISC. So I think it is true. So we are correct in the first one, second by both. This is we have done wrong. The first is, let us see the fourth, by the enzymatic reaction, and by it is false. So what is the correct one? The RNAi mediated gene silencing includes the binding of siRNA to form a complex with the target gene called RISC. Okay, so what instead of siRNA we should have double stranded RNA, dsRNA. Okay, now these are the home assignment. So these are some of the questions and link. So let us see some video or animation of this. So the links are